All right, welcome everyone to the Jones Seminar on Science, Technology, and Society. I'm biased, but if there's any field that is enabling both mind-blowing science and life-changing and even life-saving technology, I'd be saying that's neuroengineering. And today, I'm really thrilled to introduce you a true pioneer and also leader in this field, Professor Kevin Otto from Purdue University, where he's currently the Dane Miller Head and also full professor. As introduction, Professor Otto got his bachelor's degree in 1970, I believe, from Colorado State University, and then PhD in bioengineering in 2003 from Arizona State. And then he spent a few years at the University of Michigan before he launched his independent career in 2006. First at Purdue University, spent 8 years there before University of Florida brought him over. And then he spent another 10 years at Florida. But it seems that Purdue won the upper hand again and recently won Kevin over again, now as the head of the DME. I'm not sure whether there's a pattern there, Purdue and Florida, but I won't be surprised next time I see him at Florida again and maybe doing a dean or provost or something. Kevin truly is an accomplished researcher and leader in this field. Many of the practices in any neuroengineering labs, I dare say now, to name a few, electrode remediation, PEDOF-PSS electrodes, and also asymmetric electrical stimulation protocols are really invented or helped established by him. So he has certainly won numerous awards and led numerous projects as the lead PI, multi-institutional ones, and multidisciplinary ones, and also has been awarded the Society Fellows of many different communities. So I don't have the time to go through all of them, otherwise it's going to be another 10 minutes. But I just want to share a personal story before I end my introduction. As someone who actually just joined this field 10 years ago, I can say that I grew up reading Kevin's papers. And this field, just like many others, I would say without no hypes. There are always hypes. So what I like about Kevin's paper is that not only they are pioneering, they're also very rigorous, they're also very systematic. And for that, I admire Kevin all the more. So everyone, I give you Kevin Otto, and please join me in welcoming him.
That was quite the introduction. I appreciate that. Hopefully I can live up to it. It is a fun time in our field. I'll talk a little bit about that in the beginning. There's a lot of excitement. I do also want to point out really quickly, I love the idea of science, technology, and society. We, at the University of Florida, we had written a proposal for the NSF in the Engineering Research Center space, and it was neurotechnology for engineering and society. One of the issues we were looking at in particular was substance use disorder, going after could we use bioelectronic medicine or other neurostimulation techniques to help those who are suffering from substance use disorder and obviously in society, huge opportunity with the opioid epidemic. So it is true, the nervous system has big roles in this, and engineers have big roles to play. So it's a really nice aspect that this seminar addresses those things. Today I'm ultimately going to span existing projects and where we've been all the way to bioelectronic medicine at the end. Routinely, this talk is really long, so I'll try to go fast, and I apologize for the speed. But I do have to put the Purdue plug in there as the head of the school. I think I'm biased, who said but I think it's a pretty great place. We do it almost exactly the opposite of Dartmouth. Everything that we do is big, really, really big. 600 total students, 225 grads, 150 faculty. Last year, we topped 20 million in research expenditures, which was really exciting for us as a school. And we have some really nice big programs. We have an integrated MD PhD program that is statewide, so we're able to work with IU School of Medicine in this space. I am jealous of the medical center that you have right next door, but we're working in that space as well, trying to. So I throw this up there. If you're a graduate student, you may consider a postdoc, or maybe even a job someday, or maybe you're an undergrad here, reach out to me. So if I take our papers and put them into ChatGPT and ask for a word cloud, this is what we get. And that looks about right. A lot of cortex, a lot of recording, a lot of stimulation, lots and lots of different little words in there that I'll take you through today. They'll mainly start in the brain. Then we'll talk briefly about the spine and nerve, and then we'll end with a bioelectronic medicine focus. I'll bring a little bit back to my personal journey and talk about the hot field of bioelectronic medicine. So brain microdevices. As we mentioned, I've been in this space for a long time, putting things in brains and trying to understand what we can and can't do with them. And so if I have to grade brain microdevices, I would say they are good brain interfaces. And for those of you who have applied to anything in the NSF, you know that good is not very good. There's at least 2 grades better, right? Very good and excellent. So good, what are they good at? Well, here's an example that many of you probably have seen before. This individual is paralyzed in an accident, and they implanted electrode arrays into the primary motor cortex. The recordings from the primary motor cortex are used to drive the robot. So they're thinking of moving their arm, and the robot is moving. That had been done as early as 2007, at least in the literature. So this was not new when this was put out. What was new, for the first time, this individual also had microelectrode arrays in the somatosensory cortex. So when President Obama touches that robot right there, there are force sensors in the robot, and that force transduction is translated into a direct electrical stimulation of the somatosensory cortex. Quite literally, he's feeling that touch, okay? So we've learned a lot from this and experiments like this of how important the sense of touch is for embodiment and use of these prosthetic systems. Having that greatly empowers these people to use these technologies to the point that before the devices are sensorized, they say that arm that I use. After the devices incorporate sensory activity, they say my arm. There's this huge aspect of that's mine. I feel it. So we know that it's very important to do this. If we think about where the field sits right now, if I ask my parents what they know about it or other people, they say this, right? Everybody knows this. One thing that I find sort of fascinating, if I think about what happened in the world that I graduated in 2003, there were not very many job opportunities out there. I could not move to San Francisco or Austin and work with Elon and the team. Your world is very different. If you're a neural engineer coming out now, you could work with big companies. You could work with startup companies that are doing bioelectronic medicine. You could work with research-grade companies. You could work with other research-grade activities. OpenBCI, if you don't want to make any money, but you want to do it for the real reason for doing it. Other startups which are in this space, and many, many more. Right? This is not comprehensive. Everybody's taking the picture of this. Don't take the picture yet. I mean, you can take a picture of this. If there's any picture everybody takes a picture of, this is it. So take a picture. But where I will also point you back to is this fantastic website where they're doing online data-driven scraping to show you all where you can go to go look for jobs, right? This is incredible. It's an exciting time for our field. And everything that's coming out of Neuralink is fantastic, and it's just the beginning. So, getting back to micro devices, if in fact you're injured on the way home, you have a spinal cord injury, neurodegenerative disease, you cannot get this prescribed. It is not a clinical indication yet to have these devices inserted into your brain. So what's the problem? Well, us and many others have demonstrated for a long time that these microelectrodes implanted into the brain result in a reactive tissue response. The body responds, just like it would any other biomaterial. And in fact, I was at a meeting, brand new assistant professor in 2007, and Buddy Ratner came up to me and said, "You're not special." I thought, wow, that's awesome. As an assistant professor, there's nothing you want to hear more than that. And I said, okay, cool, tell me more. And he said, you all think that the brain is some special place, and you can put electrodes there, and it's not going to be just like the rest of the body. Well, I have news for you, it is. And so he said, we've published many papers on it. You're going to get multinucleated giant cells that form due to encapsulation due to frustrated phagocytosis and other cells that are coming in trying to phagocytose the device. He said, go find it. And I said, okay. So the theory is that you put the electrode in the brain and you have cells that come in and encapsulate the tissue. And we had known this for a long time. We knew that the cells were initially microglia, and later there were astrocytes that would come in and form tight junctions surrounding the device, or at least that's what was thought. David Dell and others had shown that there could be a kill zone surrounding the device if the encapsulation was toxic enough. And so we set out to try to go find it and see if we could actually look to see what does it look like. So we put Michigan electrodes into the brain and then did histology with the electrodes in place. We called it device capture histology. And I say we, I really mean Andrew Woolley did this work. He sectioned the tissue so that we could actually keep that intact interface. And then it was thick enough that he could stain using immunohistochemistry. This had only been implanted for a week. So I'll tell you that it looks a lot different if you look further out. But at a week, you can see here, lots and lots of microglia have attacked the array, just like Buddy Ratner told us that they were. In the periphery, it's macrophages, but in the brain, it's largely microglia. And since we actually have a device that translates across the blood-brain barrier, it's impossible to discern between them. But importantly, if you try to pull these apart with computer vision or otherwise, you can't. They are fused together. They're multinucleated giant cells, just like Buddy told us that they would be. We also looked at the astrocytes coming in, and you can start to see them starting to turn. If we look many weeks out, they start to form these tight junctions like what you see at the top or near that blood vessel where they're forming part of the blood-brain barrier. And then, again, as early as 2011 when we published this, we saw oligodendrocyte degeneration surrounding the device. We saw a demyelination occurring in those deeper sections. You can kind of see the lack of the cyan color. So this gave us, as engineers, this gave us a design target. This gave us who was playing what role at what time so that we could try to design devices to go back and get after that. So we've done lots of different things in this space, and this talk is not that talk. What we know is that they do have a limited lifetime, and if you're interested, we've published probably 3 really good review aspects historically of what happened before 2014. In these review articles that we wrote and book chapters, our biggest conclusion from retrospective data analysis was that it was primarily mechanical. Size and stiffness were the two most important things in terms of having long-lasting devices. This made sense from the tethering perspective. It made sense from large versus micro. It made sense from stiff versus polymeric or other software devices. It doesn't explicitly mean you can put something that causes a material that causes a major immune response. Pro-inflammatory, if you take a very irritant material, then the material becomes the biggest factor. But if you stick to something that is relatively inert, then mechanics are where you need to focus. So one other thing that we tried early on, which was mentioned in the introduction, was actually something the NIH told us to do. There was this finding that they had at the NIH where they could apply electrical pulses to the electrode, and it resulted in improved signal-to-noise ratio. And our labs at Michigan were performers on the neuroprosthesis program, so they said, you work for us, figure this out. So we did a study where we did electrolytic lesioning of this glial encapsulation. Specifically, the idea was, and it's actually, it sounds crazy, but it's not that bizarre. They actually use it for submarines. For a submarine, they electrify the windshield and all the muscles fall off. As the muscles are all stuck to the windshield and you can't see, when you electrify the windshield, it breaks all the proteins that the muscles are using to stick to the windshield, and then they all fall off. So that's one theory, that maybe if we apply, if in fact there are neurons that are still local, but they're just hidden behind the reactive tissue response, if we apply that, maybe we can see through it. And so I'm not going to dig into the impedance spectroscopy. We talked a lot. I know that there are some of you who are really interested in this, and I'd be happy to talk more. But we were able to show that with an impedance model, we're able to lower the impedance, which is associated with the reactive tissue response. As it builds up, the impedance of the device goes up. And more importantly, we were able to take electrophysiology from the left side and turn it into electrophysiology from the right side. So you could actually use this to drive a wheelchair or move a robot. You can do it over time. Every day we'd come in and we'd measure the the signal-to-noise ratio of electrophysiology, and it would be low, and then we'd do the rejuvenation, and the electrophysiology would go up. So you can, in fact, do it repeatedly, but you will note, or I'll draw your attention, this is not a flat line. It's still going down. This is not a silver bullet, at least as it stands. We're still working in this space. If peer reviewers will ever support our work, we'll have a grant to go look at this in more detail. Okay, so let's talk about microstimulation, though, because that's really my passion. This was my dissertation work, and it's where I've spent many, many years understanding it is a very powerful information transfer mechanism. The individual shown in the slide, Nathan, could use it to feel that robot, and we've been able to stimulate visual cortex to create vision, or at least small percepts. Auditory cortex, you can hear things that are not there. What have I learned from doing this for 25 years? We've published lots and lots of different things with materials and sensation, if it's safe. I'll draw your attention back to 5 primarily things. One, we know that the reactive tissue response that we've shown before, it is a factor. It does matter. Historically, the reason I got into stimulation is because you can always just turn stimulation up. If the tissue response gets bigger, you just turn it up, right? And then you can get past it. That only works for a little while. And so we needed to still go back to those design principles. There are things that we can do in this space, design-wise. And the first one was we wanted to know what depth in cortex should we go to. What's the best depth as far as reactive tissue response goes? So Morgan Urdanetto was a PhD student who did this work, and this is threshold. We were stimulating the animals. They were behaving and telling us what they could sense and what they couldn't. And what you can see here, over 40 weeks post-implantation, if we get down to layer 5, it's nearly stable. That actually corresponds really well with our reactive tissue response data. The reactive tissue response is much more aggressive towards the surface of the cortex. And so that gives us a good place of where in the brain to put the stimulation. But how about where on the device? So Hugh Lee was another PhD student in the lab who studied the idea of moving the sites around on the surface of the substrate. For whatever reason, when the Michigan electrodes were developed and others, the electrodes were all put down the middle, right? As a fabrication engineer, I can appreciate that that's where you have the most dielectric on both sides. It also turns out that's the worst place to put the electrode on the substrate from a biology perspective. So if you see the device up on the top right, we designed a Michigan electrode that had sites down the middle, like the classic device that has been around for 30 plus years. We also put a site at the tip, so it was more comparable to the Utah electrode array, the BlackRock array. And then we put these sites on the edge. And this was really informed by and using the Patrick Tresco model that showed that the reactive tissue response was the greatest at the center of the device. So this is directly from Patrick Tresco's model, showing that the dying and degenerating neurons, I lost my mouse. Well, you can see it there. Red is bad. So red near the surface of the sites, which is where we had always put them, that was the worst possible place to put them. And what Hugh found was, in fact, that the electrodes that were at the edge were, in fact, they performed much better in a recording sense. So naturally, that's ripe for an undergraduate to say, well, why didn't you stimulate? And I said, I don't know. Hugh was busy. So she picked them up. Ramya picked up these electrodes, same exact electrodes, and did a stimulation study in somatosensory cortex. We published this paper in 2021 with stimulation. And if, in fact, this is similarly impacted, the edge should have lower thresholds. They should be closer to the neurons, and they should have lower thresholds. And in fact, they are for most of the depths that we looked at. So we're still starting to learn lots and lots of the best ways to design the devices and where to put them, maybe how to stimulate. One thing that we knew for sure from our retrospective assay was that we needed to make the substrates as small as possible. And this had been in the literature for a fabrication, histological result, and recording. The fabrication and histology was done in 2007, not by us, but they showed that ultra substrate devices, things that were smaller than 10 microns in 2 of the 3 dimensions. That's our definition, but it seems to hold. So less than 10 by 10, and you still get the wire coming out. You don't have to be wireless. And that worked for John Seymour when he did this study in 2007. T.K. Kazai then in 2012 published the Nature Materials paper with carbon fiber electrodes that are ultra in 2 dimensions and showed really nice long-term stability. Okay, so now we're starting to figure out how we can design the whole devices. What should the substrate be? What should the site look like? Where should we put it? How small can it be? But we want to stimulate. And if you're going to stimulate with ultra small sites, we're probably going to have to stimulate more than one at a time. That was the thinking. So Nick Koenig, who's currently a PhD student at Pitt, he was an undergrad in the lab, and he did a study to see what happens if we stimulate 2 of the Michigan electrodes at the same time. We know what the volume conductor looks like for a single electrode. Current spreads out in space, volume conduction, and the voltage drops appropriately. And we did models to show that if we stimulate neighboring electrodes, you do get some overlap, and at some point it pulls apart, and there shouldn't be an effect of stimulating at the same time. And so these are the behavioral results. The single dot at 0, at 1, that's the normalized threshold if you stimulate with a single electrode. And if you stimulate with 2 electrodes that are separated by 100 microns, we actually saw a 47% reduction in each individual site. So let me say that a little more clearly. This is not necessarily more efficient. We're actually stimulating with 6% more total current. We have about 6% loss, but each individual site is only carrying 53% of what it was before. So it's lower on each individual site. It's safer for the individual sites, and we're getting summative stimulation. And in fact, as we pull them apart, and you get further and further apart, that effect starts to go away. Okay, so using all of that as a background, we wrote a proposal for the Brain Initiative in a UO1 program to try to do something about it. To take all of this comprehensively to say, okay, we think we know how to design this. And 2017, this was like right at the time that the Star Wars movies were coming out, so, you know, the rise of the ultramicroelectrodes. It was set up in 4 generational development stages. We designed electrodes that were going to come out with G1, generation 1. In this case, all we were doing was trying to use high-density electrodes because the electrodes that we tested stimulating together, they were still 100 microns apart. And that's pretty coarse. We thought, you know, neuropixels are basically right next to each other. So we just wanted to take a basic neuronexus electrode, the basic Michigan electrode, and put the electrodes closer together. Super simple. We know we can do that. Key to milestones is make sure you can hit them. So G1 electrodes were simply meant to test 2 things. 1, the distance between them, and 2, how small is small. So the big electrodes here have the same surface area as the BlackRock arrays that are in the humans. These are the clinical standard. The small electrodes go all the way down to 5 microns in diameter, which I thought was ludicrous small. Right, that should be way too small. And it's an optimization, so hopefully we were going to get too small so that the optimum is somewhere in there. From the foreshadowing, you probably know the answer, but that's what we tested. How were we going through it? It's a big team. NeuroNexus we design the devices, NeuroNexus manufactures them. Stuart Kogan helps us by changing the site chemistry of the devices so we can get enough charge carrying capacity through these ultra small microelectrodes. One of the powerful techniques we use to try to assess device performance and health is electrochemical impedance spectroscopy, and Mark Erasm is a very, very strong collaborator to help us with that. Warren Grill and his team help us with understanding what happens after the current leaves the electrode. So doing all of the modeling in the brain using inspired models that have real neurons that tell us how the stimulation patterns exist in the brain. And then my lab does the in vivo validation of the whole thing. So 2 taglines from this project were we wanted to understand every coulomb from amplifier to action potential. We wanted to know how it's coming from the amplifier through the electrode interface, through the reactive tissue response, and out into the extracellular space to the action potentials themselves. Ultimately, we were trying to bring, for those of you that are in the field, you may know the Shannon model. Bob Shannon wrote a paper in 1992 that limits the amount of current you can put on an electrode for neural stimulation. It was written in 1992, and that was never what he intended to do with that paper. So what we were trying to do is bring the Shannon model into the 21st century, to try to put it in so that we could have different materials. His model has no material in it. There's no variable for material. It was not drafted with microelectrodes in place. So there's a lot of updating that needs to happen. I'm not going to talk about G1 design or results, but I will show you a G2 result. In this case, we learned that we were just focusing on the ultra microelectrodes, 5 and 10 microns in diameter. These electrodes we put in different bowling pin patterns. That's what Warren Grill calls them. And we put some other fancy designs in there that help with our electrochemistry, really big reference sites, buried loops and buried terminal sites. And the goal in this space was to stimulate 6 10-micron electrodes at a time and get threshold. And if that worked, then to study 5 or 4 or 3. And if that worked with the 10 microns, well, then could we keep going? And could we stimulate the 5-micron electrodes? And keep going and see if we could get thresholds out of this. So this is the data from that study. Unfortunately, it is confounded in time in that way because we actually didn't know, we weren't going to start with a single 5-micron electrode because I didn't think that was ever going to work. So we started with 6 10s, which gives us 471 square microns. Not crazy. People have reported threshold from 471 microns before. And in fact, it works. It's robust. It's highly variable. Lots and lots of variance in that graph, but we can get threshold from that. So drop one of the electrodes and go to 5 or 4 or 3 or 2. And in fact, we start to see this curious trend where the threshold actually starts to go down. We're getting more and more and more efficient as we start to drop the electrodes. We knew that from the Koenig studies that adding electrodes makes things less efficient. But at some point, I thought that our voltage excursions, we were going to start to exceed what these electrodes could do. And we're not even close. Because we're gaining so much efficiency, we have a lot of headroom on the design space of the electrodes. So, interestingly, we start to cross over and 6 5-micron electrodes and 5 5-micron electrodes all the way out to, you start to see the error bars get really small. Elizabeth Olivo is in my lab now. She's doing this work. And we were just at a meeting last week at BMES where she absolutely threw me under the bus, which is fine, because she would have more data here, except for she had to move the lab from Florida to Purdue. So we have this data. It's remarkable. It shows us the answer that we needed. It's not published yet, so you have to take a picture because it's not out there because we're still tidying up more data in the new location. But this is a UO1, it's milestone driven, so we have to keep going. And so G3, the big change here is to start to shift to the ultra substrate. Shifting to a new substrate material, silicon carbide, that we can start to make the substrate as small as possible. The previous substrates were on the order of 20 to 30 thickness, which is too thick for the ultra capacity. So getting down to a 5-micron substrate. In this study, what Elizabeth has done is simulate with various versions of 20s, various versions of 15s, 10s, and 5s. And I'm not going to belabor this too much because I kind of want to get to the later parts of the talk. But so far, the results that we saw in G2 are extremely consistent with G3. As we continue to progress down and further in number of electrodes and in size, our threshold is getting smaller and smaller and smaller, and at least at this point, we haven't figured out the optimal size yet. We haven't quite miniaturized as far as we possibly can, which is pretty exciting. I thought we would have hit the minima a long time ago. We have lots of different animals who are able to do this, and the data are actually incredibly consistent. Okay, so just for fun, we have no data from this yet at all, but the G4 electrodes, this is our year 4 of this program, and G4 are supposed to be the final design. Ultra substrate, ultramicroelectrodes in the locations that we want. And this is the design that we chose. This is what is now coming out of being fabbed right now. It's just rolling off, should be packaged and actually waiting for me next week when we get home. So stay tuned. I'll tell you the answer of the G4 study eventually. Okay, so let's shift gears just a bit. I want to talk briefly about some of our work in spine and nerve modulation. And honestly, this is because I want to make sure that the graduate students are represented. They've worked hard on this, but also B, to point out how important the collaborators are in this process. Our first work here was a big collaborative team that was largely led by Jack Judy at Florida in collaboration with Christine Schmidt and Carlos Rinaldi. So we published this idea of a tissue-engineered neural interface. The goal here was to have Jack build the devices in the clean room, polyimide threads, multi-site electrodes, embed them in Christine Schmidt's hydrogel that's then encapsulated like a burrito with small intestinal submucosa as the tortilla. And then my lab would take a chunk of nerve out and suture the device in place to see if the nerves would grow back through this tissue-engineered construct to give us recordings and potentially be used for stimulation. So this is what the devices look like. I mainly show you this because light sheet microscopy is super awesome. But what Eric Atkinson, who did this work, what he was able to show is that the regeneration through the devices and around the devices was robust. And in fact, he was able to get really, really strong electrophysiology from these regenerating neurons. He asked me a few times, is this what this is supposed to look like? And I said, I don't know what regenerating axons look like. I know what normal axons look like. They look like what I think they should look like. But yeah, this paper was published 3 years ago. So we've also done some work in spinal cord, primarily epidural stimulation of spinal cord. And where our focus in this has been is with a strong respiratory collaborator, Erica Dale. We did closed loop stimulation for spinal cord and breathing. That actually turns out to be remarkably easy. There's so much cool stuff right now coming out with epidural stimulation. Onward is doing phenomenal stuff, and it just it turns out it's remarkably easy to stimulate the surface of the spinal cord. I'm not trying to downplay what they're doing. That's super cool. But and then Marco Crapogrosso at Pitt with their stroke recovery, really, really exciting stuff there. We've also done a lot of work on in situ development. I know there's a lot of interest in PDOT here. PDOT can be polymerized inside the body. It's a little tricky because the monomer is toxic, so you can't do it the typical way. You inject the monomer around the electrode in situ, and then use electric energy to polymerize it. So you can do spatially controlled PDOT deposition both in the brain and in the peripheral nerve. It's really exciting, fun stuff. Okay, last part of the talk, bioelectronic medicine. This one is really fun for me because it's really close to home. Some of you have already heard some of the story, but I'm a very transparent, direct person. This is a graph of my body weight for 5 years. Something like that. I like to mess around with diet and exercise and take data. I'm not this crazy. We have a Fitbit ARIA scale that records the data all the time, so I don't write it down every day. But you'll note that most of you probably don't have a point in your life where you lost 80 pounds. In November of 2013, right there. That was the point where Jack Judy talked to me at the Society for Neuroscience meeting about moving to Florida. And I'm not saying that Jack did this to me, but there was initial indications that I probably had some things going on with my pancreas and otherwise, but that stress of thinking about moving the lab, it's a big thing. But it was all hidden because it was January 1st, so you probably cut down on sweets a little bit after the new year, and we were training for a mini marathon, and it was the best diet ever until at some point I'm like, wait, I'm not really even trying, and I'm still losing a lot of weight. So I lost about 80 pounds, and when I'm now recovered. It turns out I was diagnosed with adult onset type 1 diabetes. My partner is a lab animal vet, and so for any of you that do lab animal work, now that I'm recovered and it's a fun joke, I can say, she jokes that I probably should have been euthanized because I had lost more than 20% of my body weight. But, this was pretty bad, right? I was in the ICU for 24 hours, and insulin is a wonder drug. And having access to insulin was really remarkable, recovered the weight pretty fast. But it was a really interesting time. I hadn't given much thought. I was very much an elitist about the brain. I did not care about even the brain stem or hadn't done any of the spinal cord work. Couldn't have told you what the difference between type 1 and type 2 diabetes was. In this time, 2014, the lab is getting ready to move. And this whole idea of bioelectronic medicine was really starting to come out. This editorial was written by Kevin Tracy in June of 2014 in the inaugural issue of Bioelectronic Medicine. So if you're the editor, the founding editor, on page 1, journal 1, you get to write, what is this journal about? And that's what this article was. Kevin wrote this article June 2014, and the lab moved to Florida August 2014, which also means I had a whole bunch of startup money that we could go and do whatever I wanted. And it also turned out I had some really, really great diabetes collaborators there. So we started to look at this idea of pancreatic neuromodulation. I had a lot to learn. Like I said, I didn't even know what diabetes was really, but I had a vested interest in learning about it. And so the first thing we did was map some of the innervation. It is true, every cell in your body is influenced by your nervous system or talks to your nervous system. And in fact, if that neural innervation goes away, a lot of times the cells die. They're pulled away from the brain. They don't understand what's going on. They die off. So we mapped, it is vagal innervation that comes down and goes into the pancreas. It actually interfaces directly into the beta cells. We published that in 2018. We then set off on a study to see if we could do selective neuromodulation of the pancreas innervation. This was really hard. We also weren't the first people to come up with this. People had tried it before, but they were always stimulating proximal to the liver. When the vagus nerve comes down, it goes into the liver and transverses through the liver and then comes out the other side. In the meantime, it's innervating the liver, and then it goes into the pancreas. So everybody was stimulating right before the liver, and when they stimulate there, they're stimulating both the liver and the pancreas. The liver is involved in raising blood sugar, and the pancreas is involved in lowering it. So they're basically hitting the gas and the brake at the same time, and publishing lots of papers that say this doesn't work. So you have to stimulate the nerve after it comes out of the liver, which is not an easy thing to do. In their defense, we honestly didn't have the devices to do it 20 years ago, 30 years ago. We partnered with a company, Qualia, to develop shape memory devices that could wrap around the innervation of the pancreas. And I say we, I really mean Elliot Durr did all of this work. It was heroic. He put continuous blood glucose devices into the rats, induced type 1 diabetes in the rats, and showed that even in a type 1 diabetic model, he was able to stimulate and result in insulin secretion in these animals. So it was a tough experiment. It's far from optimized for sure. There's a lot of work that needs to go into potentially extending this further, but it did show proof of concept. We've done some other bioelectronic medicine work in pain modulation, which I would argue is extremely important. If any of you have spare cycles or you're early in your career, this is an area that I think is incredibly important, a lot for the society that I mentioned earlier. Trying to have alternatives to drugs is really, really important. It turns out that the neuro-gut axis is really important for something like hypertension, and we did a paper here stimulating the vagus nerve in a hypertensive rat. And we had a long DARPA program stimulating the vagus nerve for learning. The vagus nerve also goes back to your brain. And so I joke a lot that to learn something, you need to be in a good parasympathetic space. I make all of my classes breathe deeply before we take tests, because if you're freaked out, your brain doesn't work. because your sympathetic nervous system is dominant. And so what we've shown in this learning area is that by stimulating the vagus nerve, you're pushing people into a parasympathetic learning open space. Last thing, I'm big on collaborative team-based science. You have outstanding people here, so you have a lot, and I've heard a lot of team-based activities, but this is a little bit of a play to the people who aren't in this room, who need to hear it, that, you know, we need to fund large team-based science because that's where these intersectional fields like neural engineering are accomplished. And so much of our work has been done by big teams of various investigators that bring in lots of different disciplinary ability. And I was challenged by a DARPA program manager in 1999 not to throw things over the fence. Don't just do your part and then hand it and run away, right? If you don't know how to do something, hand it to them and then watch them do their part. Because that really helps with, so that you do your part better, so that they can do their part better. So with that, I will say thank you for your attention, and I'm happy to take any questions.